so welcome to my hot wash video. So for this unit, I'm going to be talking about all the mitochondrial transporters that we went over. And it really is just like a hot wash, just trying to get them all straight in your head. So let's start out with the aspartate malate transporter. So the purpose of this transporter is to transport NADH into the mitochondria by using aspartate and malate and the reactions that take one from the other. So just to orient you a little bit, here's the cytosol, here's the mitochondrial matrix, and here's what we're looking at, aspartate. So aspartate can be turned into oxaloacetate, which can then be turned into malate, and in that process, the malate gets reduced and which oxidizes this NADH back into NAD+. The malate can then go through a shuttle into the mitochondrial matrix, where it can then reduce NAD+, that is inside of the mitochondrial matrix, into NADH, and then bam, now we have NADH to use in oxidative phosphorylation to make ATP. And then the malate can go back to oxaloacetate and then back to aspartate, and then it can go back and repeat the cycle. Okay, so the next transporter that I want to talk about is how fatty acids come from out of the cell, then into the mitochondria. And that starts with activation. And that whole process starts with extracellular hydrolysis. So there's this enzyme that's on the surface of your cells. It's called lipoprotein lipase. It's a lipid linked to your membrane, and it's usually in the walls of blood vessels. What it does is it hydrolyzes TAG into glycerol and fatty acyl groups as it passes by, and it can create 3 fatty acyl groups from this. So it takes this triacylglycerol and it hydrolyzes it into a diacylglycerol, and then into a monoacylglycerol, and then it can also release that last one and releasing fatty acids in the process. And then from there, these fatty acyls can be taken up into the cell by a transport protein. So from there, before it can come into the mitochondria, it needs to be activated. So the fatty acyl groups are activated by ATP, and as a result of that, it releases 2 inorganic phosphates. So as you can see here, the fatty acyl group attacks the ATP, it grabs a phosphate for itself. So the resulting, it's called an acyl adenylate. It reacts with a coenzyme A, which displaces the AMP, and so now we're left with an acyl CoA and an AMP, and from there that acyl CoA is now going to be able to use the carnitine transporter to get into the cell. Which brings us to the carnitine transporter. So this is responsible for transporting activated acyl CoA groups into the mitochondrial matrix. So this is how it works. So this is us in the cytosol, and we have this fatty acyl CoA that's been activated. So what the carnitine does is it displaces the coenzyme A, and it grabs that fatty acyl group, and bam, now you have fatty acyl carnitine. And then from there it can go transport through the carnitine transporter, come out the other side, and then the CoA again can go displace the carnitine this time and reproduce that fatty acyl CoA. And then we can go through beta oxidation, create acetyl CoA, and all that good stuff. Okay, so next is a citrate transporter. And what is the function of the citrate transporter? So what it does is it moves citrate between the mitochondrial matrix, where it's made by the citric acid cycle, and into the cytosol, where it can be made into oxaloacetate, into acetyl CoA, and then from there into fatty acids, cholesterol, and triacylglycerols. And it's also an antiport with malate, so as the citrate moves out, the malate can move in and become used in the citric acid cycle. Okay, next is the citrulline transporter. And so the citrulline transporter, this is what transports the citrulline out of the mitochondria into the cytosol during the urea cycle. And the way I try to remember this is citrulline, cytosol. Citrulline is going into the cytosol. And that comes after that first activation step where you make the carbamoyl phosphate, and then the carbamoyl phosphate combines with the ornithine into citrulline, and then it leaves the mitochondria and goes through the rest of the cycle. Which brings us to the ornithine transporter. So the ornithine is going to be made inside of the cytosol, and it's going to be needed to transport it back into the mitochondria so that it can be combined with the carbamoyl phosphate into the citrulline. And also, interestingly, it can also transport lysine, arginine, and citrulline as well, which I thought was interesting. Okay, so now let's hit the ADP and ATP transporter. So this transports ADP into the mitochondria and ATP out. So basically it's replenishing the ADP supply so that it can be used by ATP synthase to make more ATP. So here's the intermembrane space, here's the mitochondrial matrix, here's ATP synthase, and here's the transporter that we're looking at. So what it's doing is it's moving ADP in, and then from there it can combine with a phosphate that's brought in by our hydrogen phosphate symport to create ATP, and then that product will be able to move out. Which brings us to the hydrogen phosphate symport. So this is what's providing that phosphate for ATP synthesis, and it's basically, if you remember, throughout this whole process, all these complexes are pumping these hydrogen into the intermembrane space in order to create this gradient so that ATP synthase can use that gradient, that protomotive force, to spin itself and to combine ADP and phosphate. So this symport takes advantage of that, and it uses that gradient to push hydrogen in, and using that energy, it also can push phosphate in. And this is just saying the same thing essentially. Yep. And those are all the transporters, and here are my sources.